Hi, everybody out there on the internet. My name is Denman, and I'm a master's student at the University of Calgary. And I'm going to be talking about my master's thesis, which has the running title Applying Genomic Selection to Improve Pacific Oyster Chrysostria gigas Resistance to the Pathogen Vibrio aestuarianus. I'm in the Department of Biological Sciences at the University of Calgary, and I'm co-supervised by Dr. Sean Rogers at U of C and Dr. Tim Green at VIU. So, a little bit about my background. I did a diploma in fisheries and aquaculture at VIU. I then did my Bachelor of Science at VIU, and I'm now doing my Master's of Science at U of C. I primarily work out of the Deep Bay Marine Field Station, doing a lot of my field work, raising my oysters, and then the Center for Shellfish Research on the VIU campus, doing my lab work and a lot of my data analyses. So for my project, just going over a little bit of the background information, I'm going to start talking about Vibrio aestuarianus, or VA, I'm going to be referring to it as. So many people watching this probably already know about Vibrio, but essentially it is a bacteria. It is a well-known pathogen of Pacific oysters and has been heavily correlated with our summer mortality events, which are our marine heat wave events that we're getting more often every summer on the BC coast, and these summer mortality events are causing losses of 20 to 100% of farmed oysters in BC on different farms. So we wanted to ask what we could potentially do about this. And so something that we know has been successful in the past is selective breeding for disease resistance. So we've seen this sort of across the board on different species and then also against different taxonomic groups of pathogens. So we've seen it be successful against fungi, against viruses, and against bacteria. And at the bottom of the screen for the shrimp example against early mortality syndrome, selective breeding was actually successful against another species of Vibrio, so Vibrio parahemolyticus. So we do know that selective breeding can be successful for breeding for disease resistance. For my project, we wanted to ask what type of selective breeding would be appropriate. So there's sort of three main umbrella terms of selective breeding. So the first one is family-based or pedigree selection. Essentially, this uses no genetic information. It just uses a pedigree of who is related to who up the family tree. There's also marker-assisted selection, which is used for when you have a trait that is controlled by one specific genetic marker. So this does use genetic information, but it's only applicable in certain scenarios. And then the third one that's becoming more common as our genetic technology sort of increases over the last couple of years, this has become more popular, is genomic selection. So genomic selection uses high density genetic data, so like large segments of the genome. It is ideal for traits that are controlled by many different genes, and resistance to Vibrio aestuarianus is suspected to be a complex trait that is controlled by many different genes. So this is the type of selective breeding approach that we decided to use for my project. So the problem with breeding for disease resistance is that how do we actually know how resistant an oyster is to a disease? We need to do a disease challenge. So essentially, you give the oyster the pathogen and you see how long it takes for them to die. So when they have a very fast death, they have a low resistance. When they have longer survival, they do have a higher resistance, and that becomes your phenotype or your trait. But the problem with this is that it is destructive sampling. So when we know the oysters that have the trait of interest, by the time we know that, the oysters have been destroyed during that process a lot of the time. So either they have died during that process, died from the pathogen, or they have been so heavily infected by the pathogen that you wouldn't want to use them as broodstock, or they're likely going to die very soon after the trial. So the solution to this, and where genomic selection comes in, is that we can use the genetic information of the oysters that went through that initial disease challenge, and we can compare that to the siblings that are still alive. And using that, we can identify the best individuals within that sibling group to produce the desired trait. And essentially, your sibling group then become the candidate broodstock for the next generation. So for my project, one of the first things we had to do was figure out how resistance varies between different individual Pacific oysters from different genetic family backgrounds. So we had to do that disease challenge using Vibrio aestuarianus. So this was done back in 2024. We had 19 different oyster families. I'm not going to get into a ton of the details of the disease challenge, but essentially you have one little oyster inside each well of a big well plate, and you give them the bacteria and you see how long it takes for them to die. So we had our oysters in a temperature controlled room at 23 degrees to try to simulate a marine heat wave. And we watched them for 7 days. When the oysters would die, we would preserve their tissue in ethanol. And at the end of the trial, all of the oysters got sent off for DNA sequencing. So at this point, we had our genotypes of the original oysters that went through the trial, as well as our phenotypes, so that disease resistant trait. And we could move forward with the project from that point. So this is just a very simple bar plot showing the survival that we saw during that disease challenge. So on the left axis, we've got percent survival, and then on the bottom, we have our 19 families. And really, all this is meant to show is just that there is a big difference between percent survival between different families. So in our highest surviving family, we have about like 48% survival, and in our lowest family, we have about 4% survival. So this is what we are trying to essentially hijack and try to breed for. So at this point, we have our phenotypes and our genotypes. So what is the next step? Well, remember that we want to use the genetic information of the oysters that went through the disease challenge originally, and we want to compare that to the siblings so that we can identify those ones in that group that have the phenotype that we're looking for. So how do we do that? Well, we need to collect the DNA from the candidate broodstock or the sibling group. So this is another issue with DNA sampling is a lot of the time you take tissue and you send the tissue off, but a lot of that time that process can be lethal. So we wanted to keep these guys alive so that we could breed them. So we needed to do it in a non-lethal way. So how we did this is we took all of our oysters and we sedated them with magnesium chloride, which is just Epsom salts. They get really drowsy. They let go of their adductor muscle. They open up and we were able to just collect their DNA by swabbing their gills very gently. I then extracted all the DNA from those swabs. So we had 440 oysters that went through this process, and we sent all of that off for DNA sequencing as well, which gave us the genotypes of our candidate broodstock. So the next step was to actually decide which of those candidate broodstock to use to breed our next generation. And how we did this is we calculated estimated breeding values. So a lot of the people watching this, I'm sure, know what these are, but essentially they are a numerical value. They predict the genetic merit of your individuals, and that allows you to identify the broodstock that are best suited to produce your desired phenotype, which in this case is disease resistance. So this is done using a linear model called a BLUP. I'm not going to get into the math behind the model, but essentially you supply this model with your pedigree, so who is related to who across generations. You have your phenotypes, so this would be the survival of the oysters that went through that initial challenge, and then you have your genotypes of your disease challenged individuals as well as your new candidate broodstock. And one of the things that this model outputs are estimated breeding values. And then using those estimated breeding values, you can look within your candidate broodstock and find individuals with that predicted high resistance or predicted susceptibility to the pathogen. So using that information, we then wanted to go forward and do a spawn. So in August, this past August, we spawned a bunch of different families. So my experimental design was to take oysters that had predicted high survival, breed them together to create offspring with high survival. We then did the same thing, but with a highly susceptible group. And then our control was one highly resistant broodstock with one highly susceptible broodstock, breeding those together to try to make a family with moderate survival. And then the next step was to do another disease challenge. So if there's anybody out there that saw me in October at the AGM, I said I was going to do this in November. Had some issues with that trial because our room wasn't cooperating with the temperature, so I had to redo it again in January. So I did this in January. So essentially, we took all of my families, we gave them the bacteria again. There's about 1,200 oysters in this challenge. And then my predicted results I will get into in just a moment. But this is what that trial looks like in real life. So you have all your different little well plates. In each of those wells, you have one individual spat. The controls got autoclave seawater. The ones in the actual challenge, your treatment group get the bacteria, and then every single day for 7 days, I would go in and look at them and see which ones had died. So my predicted results, I was really hoping to see that my high survival group would in fact have high survival and my low survival group would have low survival. And this was meant to evaluate the success of the project. Um, I did not see exactly this. I did get some really interesting patterns that I'm sort of working through at the moment. Um, and if you watch my final thesis defense, I will hopefully have more information for you at that time. But for now, I can say I did not see exactly this, but I still got some really interesting results that I'm excited about. So the big picture for this project is we wanted to try to selectively breed for disease resistance and just asking the question, can we breed for resistance to Vibrio aestuarianus in Pacific oysters using genomic selection? And ideally, this is meant to increase survival during the summer mortality events on our coast and bolster our aquaculture industry here. Thank you for listening. And if anybody has any questions, they are welcome to email me at this email below. Thanks.